Welcome to our today's lecture on biochemistry of nucleic acids and nucleoproteins. My name is Raymond. I'm a PhD researcher in biochemical pharmacology. So today we'll be looking at the biochemistry of nucleoproteins and nucleic acid. From our primary, we're looking at the fate and dietary nucleic acids and nucleoproteins. We'll also be looking at the novel biosynthesis of purines and pyrimidines. We'll also be looking at salvage pathway of purines and pyrimidines. We'll also look at the catabolism of purines, and let's just say catabolism of nucleic acid. We'll also go further into looking at the disorders that are associated with nucleic acid metabolism. Now, what is the fate of dietary nucleic acid and nucleoproteins? Or let's start by defining what's a nucleoproteins. So nucleoproteins are conjugated proteins with prosthetic groups of nucleic acid. So from the word, it is derived from nucleotides or nucleoside or nucleic acid and proteins. So when they are fused together, they are known as nucleoproteins. You can see the ribosome, which is a combination of the RNAs and so many other proteins. The ribosome is the nucleoprotein. Also look at the chromosome, which is also a nucleoprotein. So when they say prosthetic group, it means that the nucleic acid and the proteins are covalently linked together. So nucleoproteins are a component of every living cell. They are part of every living cell, both viruses, bacteria, eukaryotic cell, prokaryotic cell. So nucleoproteins and nucleic acid are found in foods of both plant and animal origin. So they can be found in our food. Nucleoproteins and nucleic acid, on the other hand, are not required nutrients. Why we say they are not required nutrients is because the body can synthesize them. The body can synthesize them. So when we say it is not a required nutrient, you can say it is a non-essential nutrient, since the body can synthesize them. So we don't need external source, or we don't need to take it through our diet to get the nucleic acid that our body needs for growth and reproduction. Our body can actually synthesize the nucleoproteins and the nucleic acids. So nucleoprotein digestion and absorption. So also in the mouth, the dietary nucleic acid stayed unaltered. They stayed unaltered means the body, the mouth does not have required enzyme to metabolize the nucleic acid. The dietary nucleoproteins are denatured in the stomach by gastric PHCl, which is the hydrochloric acid that is present in the stomach. The nucleic acids hydrogen bonds are broken in the stomach. The small intestine is responsible for the majority and full digestion of the nucleic acid. These are the little things that we should know. Now, the pancreatic and intestinal juice that contain specialized enzymes necessary for the digestion of DNA and RNAs, which function precisely to break the bonds. So those enzymes that are present in the GIT are responsible for breaking the bonds in DNAs and RNAs or entirely the nucleoproteins. So we have the deoxyribonuclease. We have the phosphodiesterase digest nucleic acid in the small intestine to produce nucleotides. So I would say deoxyribonuclease / phosphodiesterase. Phosphodiesterase breaks the phosphodiester bonds that is found between nucleotide to nucleotide. Now, what is a nucleotide? A student should be able to know what is a nucleotide. A nucleotide must contain the sugar, the ribose sugar or the deoxyribose sugar, the base, which can be a purine or a pyrimidine. the base. My handwriting pen is not functioning optimally, and the phosphate group. The phosphate group. So this is a nucleotide. A nucleotide must contain the base, which is either adenine, guanine, cytosine, uracil, or thiamine. The ribose sugar, which can be a deoxyribose or a ribose sugar. And the phosphate group. Actually, it is the phosphate group that connects individual nucleotides, because the DNA is a polynucleotide. So the nucleotides are the monomers, they are the monomeric units. So the phosphate group that is found in the nucleotide is what is used for the attachment. Though in our secondary schools they will always tell us the DNA is made up of the nitrogenous base, the pentose sugar, and the phosphate ladder. So what connects the individual nucleotides are the phosphate group. So that's why we have the phosphodiesterases that breaks down the phosphate group to separate the individual nucleotide that makes up that particular DNA or RNA. So we have the nucleotides. The nucleotides break down nucleotide, and nucleosides break down nucleoside. So what is the difference between a nucleotide and a nucleoside? A nucleotide contains the three major constituents: the phosphate group, the sugar, and the nitrogenous base, while the nucleoside only contains the ribose sugar and the nitrogenous base without the phosphate group. When we go further in this course, we'll also look at the definition of how we name the nucleotides and how we name the nucleosides of the purines and the pyrimidines. So we have the nucleosides break down nucleoside, while nucleotides break down nucleotides. Nucleotides and nucleosides are broken down into three parts, which is the phosphate group, the pentose, which is the sugar, because anyway you see OSE, it means we're talking about the sugar. The pentose, which is a 5-carbon or 5-membered ring sugar, which is either the ribose present in RNAs or the deoxyribose present in DNAs. And we have the nitrogenous base, which we've actually established. We also look at the digestion of nucleoproteins. So nucleoproteins in the stomach, we have the gastric acid and the pepsin. The pepsin is an enzyme. So we have the nucleic acid, which is further broken down in the small intestine by endonucleases, which are RNAs or DNAs. The ASE, you're seeing that you know that molecule is an enzyme. So we have the ribonucleases and the deoxyribonucleases. We break the nucleic acid into the nucleotides. The nucleotides will now break them into phosphate group and nucleoside, which is further broken down by the nucleosides to base and the ribose sugar. So we have the end of nucleic acid digestion, which is the nitrogenous base, which is either the purine or the pyrimidines, the sugar, which is either the ribose or the deoxyribose, the phosphoric acid, which link the individual nucleotides together. So let's talk about the absorption. So dietary purines and pyrimidines derived from nucleic acid breakdown are absorbed via the intestinal lumen. Purines that are not absorbed are metabolized by the intestinal microbia, which is called what you call the microbial flora. Microbial flora are those microorganisms that are beneficial to your GIT, and they are expelled in feces. So the absorbed nitrogenous base are carried to the liver via the hepatic portal vein. These are degraded and excreted out of the body. So as a result, the human body does not rely on dietary nucleic acid for their utilization, since the body can actually synthesize the nucleic acid. The body can actually synthesize them using the novel synthesis of purines and pyrimidines or the salvage pathway. So in this course, we also look at the novel synthesis, and we also look at the salvage pathway. We also look at some of the diseases that are associated with purine and pyrimidine metabolism. So the ribose can be absorbed and catabolized to generate energy. The ribose is a sugar which can function in the non-oxidative pathway of the pentose phosphate pathway, non-oxidative steps in the pentose phosphate pathway, and be converted to maybe into one of the 6 carbon sugars like the fructose, the glucose, the mannose, the galactose, and the rest. So that ribose that is broken down can actually be used to generate energy or ATP for cellular utilization, for cellular energy transduction. So the nucleotides are chemically composed, which I've actually explained: the phosphate group, the nitrogenous base, and the sugar. Thank you for this lecture. We'll be looking at lecture 2 in our next lecture series. Thank you, and God bless you.